Imagine you live in a house that automatically remodels itself the moment the weather gets bad.
Oh, wow. Okay.
Yeah, so like a tornado warning sounds, and suddenly the house reinforces its own walls. It pulls materials from the inside to add, thick, heavy layers of brick until it becomes this impenetrable bunker.
Right. That sounds intense.
But then say there's a catastrophic flood. Instead of thickening, the house stretches itself out, transforming into this long, elevated hallway just to stay above the waterline.
Which sounds completely like science fiction, obviously.
Exactly. It sounds like sci-fi. But the human heart does exactly this when it's put under severe mechanical or pathological stress. This architectural shape-shifting is a biological process called cardiac remodeling.
Yeah, and for decades, cardiologists have known that this happens. But the microscopic mechanics of, how the heart actually chooses its new shape, that's been a bit of a black box.
Right, until now. Because a really fascinating 2026 paper published in Science by Scarborough and colleagues just kind of blew the lid off that box.
They really did.
Our mission today for this deep dive is to uncover this exact molecular toggle they found, the switch that tells heart muscle cells, the cardiomyocytes, which way to grow. Okay, let's unpack this. The heart physically adapts in two distinct ways, right?
Yeah.
There's concentric hypertrophy, which is a thickening of the cell walls, and then eccentric hypertrophy, which is thinning and lengthening of those walls.
And we really need to emphasize the clinical stakes here for you listening, because these aren't just harmless adjustments for a tougher workout.
Right.
This is the literal difference between life and death. Concentric hypertrophy, that inward thickening, that is the hallmark of hypertrophic cardiomyopathy, or HCM.
It's the one that affects athletes sometimes.
Exactly. The muscle walls become so incredibly thick and stiff that the heart physically struggles to relax and fill with blood. It's often the hidden culprit behind sudden cardiac arrest in young, healthy people.
Wow. And the other one, the eccentric.
Right. On the flip side, eccentric hypertrophy leads to dilated cardiomyopathy, or DCM. The heart stretches out, the walls get dangerously thin, and it becomes this, flabby balloon.
It sounds terrible.
It is. It just cannot generate enough force to pump blood out to the body. So understanding how a single cell chooses to go wide versus go long, it's not just academic, it's the key to entirely new ways to treat heart failure.
And what this paper reveals is that the answer lies in something called the microtubule highway. But, testing this isn't as simple as swabbing a cheek and looking under a microscope.
Oh, definitely not.
The researchers had a super rigorous algorithm of evidence. They moved from in vitro lab dishes to in vivo living mice, and finally to actual clinical human tissue.
Yeah, and to really grasp what they found in the lab, we have to zoom way in, inside the cardiomyocyte, at the microtubules themselves.
Okay, paint a picture for us.
You can picture microtubules as these highly dynamic structural filaments inside the cell. They serve a dual purpose. On one hand, they're the steel scaffolding giving the cell its shape.
Right.
But on the other hand, they are an active, bustling internal highway system. Motor proteins literally walk up and down these highways carrying vital cargo.
Like what kind of cargo?
Things like messenger RNA and these massive protein-building machines, just delivering them wherever the cell needs them most.
Okay, so to study this in the lab, the researchers started with young, lab-grown rat heart cells. But if you just drop these cells into a standard liquid petri dish, they flatten out like fried eggs.
Yeah, and a fried egg doesn't act like a contracting 3D heart muscle.
Right. So to get around this, they place the cells on highly specialized nano-patterned substrates. This texture basically forced the young cells to mature, stretch out, and align like a real beating heart wall.
Which is such an elegant setup.
It really is. So once they had that realistic canvas, they ran the first big toggle experiment. What did that look like?
Well, they wanted to isolate what happens when you manipulate the stability of that microtubule highway, so they use chemicals. To stabilize the microtubules, like turning the highway into solid concrete, they use a drug called Taxol.
Okay, Taxol to stabilize.
Right. And to do the exact opposite, destabilizing the microtubules into a fluid, chaotic state, they used a drug called nocodazole.
And the results were pretty immediate, right? Stabilizing made the cells wider, destabilizing made them longer.
Exactly.
But there's a crucial detail here. They didn't just measure the outer boundaries of the cells, they measure the sarcomeres, the individual microscopic engine units of the muscle.
Yes, this is so important.
The sarcomere length stayed exactly the same. They didn't just stretch out like a rubber band. The cell was actively building new units.
Which is the gold standard for proving true biological hypertrophy. If the units just stretched, the heart would just get weaker. Preserving that baseline length proves it's a deliberate architectural blueprint.
Okay, so just to do a quick 5-minute recap for you listening. Bullet 1, the heart adapts by thickening or lengthening. Bullet 2, microtubules act as the toggle switch for this direction. So here's a question for you to think about. If you're designing a cell, how would you physically redirect heavy building materials from the ends of the cell to the sides?
It's a massive logistical challenge. At the molecular scale, to widen the heart, you have to completely reroute the cell's internal supply chain.
And here's where it gets really interesting. How does a stable microtubule actually make a cell wider? I like to use the SSS mnemonic here.
Oh, that's a good one.
Yeah, so it goes. Stable microtubules equal short axis growth, which means the side walls thicken. SSS.
Right.
Imagine a factory where the conveyor belts suddenly pivot, sending all the bricks to the side doors instead of the front and back. But what's the actual science behind that?
So they track this in granular detail using RNA FSH. They basically lit up specific molecules of messenger RNA. They focused on one called ACTMC1.
ACTMC1, what does that one do?
It carries the genetic blueprint for cardiac muscle protein. Now, normally, this blueprint is exported out of the long ends, the poles of the nucleus, but the moment they stabilize the microtubules with Taxol, that mRNA physically relocated.
Wow.
Yeah, it shifted entirely to the short axis, the sides.
Wait, I got to push back a little. mRNA doesn't just sprout legs. How does a strand of code actually move against its default path?
Well, that's where the LINC complex comes in.
The LINC complex?
Yeah. You can picture it as a series of heavy-duty grappling hooks and motors embedded right into the envelope of the nucleus. Proteins like KIF-IB, which is literally a motor, along with NUP153 and SUN1, they physically couple the nucleus to those stabilized microtubules. So when the highway locks down, this LINC complex acts like a rail yard switch. It actively grabs the mRNA and drags it out the side doors.
That is wild. And they proved this in vivo too, right? In living mice.
They did. They used a tiny mini pump to infuse a drug called phenylephrine, which acutely spikes blood pressure.
So simulating chronic high blood pressure.
Exactly. And the mice hearts exhibited early microtubule stabilization and the exact same short-axis mRNA shift.
Okay, time for another quick recap. Bullet 1. Stable microtubules cause concentric widening growth. Bullet 2. They do this by mechanically redirecting mRNA out the sides via the LINC complex. So for you listening, think about the structural integrity of a building. If you only add bricks to the side, what happens to the load-bearing joints connecting the front and back?
That is the perfect question to transition to eccentric hypertrophy, the lengthening of the heart.
Right. What happens when the microtubule highway is completely destabilized? Let's use another mnemonic here. DED. Destabilized equals ends grow, resulting in a decompacted disc.
The intercalated disc.
Yes. Tell us about the disc.
So the intercalated disc is this critical zigzagging junction where two heart cells connect, because the heart has to beat in unison, right?
Right, they can't just be loose.
Exactly. They are locked together by a protein called N-cadherin. Think of it as a heavy-duty biological weld.
Okay, so when they destabilize the microtubules using the cortisol or a TTL virus, what happened to that weld?
It decompacted. It loosened up.
But here's the tricky part of the science. Did the loose disc cause the cell to grow longer, or was it just a side effect? This brings us to the hacking the door experiment, which I love.
Oh, the NAA peptide experiment.
Yes. They use this NAA peptide to chemically block N-cadherin, basically forcing the intercalated disc to spring open without ever touching the microtubules.
Right, isolating the variable. And the result, the cells elongated perfectly.
That's from opening the door.
What's fascinating here is that it reveals the cell is always intrinsically poised to grow longer.
Wait, really? Always?
Always. Because the protein-building ribosomes, they naturally just hang out at the far ends of the cell. They're idling there. The intercalated disc acts as a locked door. Destabilizing the microtubule simply unlocks the door, and those idling ribosomes start adding new sarcomeres in series.
So widening the cell takes all this active LINC complex motor work, but lengthening is basically passive. Just take the brakes off.
Exactly.
Okay, recap 3. Bullet 1. Destabilized microtubules cause eccentric lengthening growth. Bullet 2. This happens because the intercalated disc loosens, removing the physical barrier at the ends. So listener, if we know how to unlock the door to stretch a cell, could we hypothetically design a drug to lock it back up in a failing dilated heart?
That is the multi-billion-dollar question.
Right. But before we get to the therapies, How did they actually see these new microscopic building blocks being added?
Right. So they used adult rat cells for this, and they executed this beautiful pulse chase experiment. They used a highly specialized TKM1 halotag virus.
Halotag, like a fluorescent marker.
Exactly. First, they blocked all the old existing proteins using a chemical called 7-bromo-1-heptanol. It essentially blacked out the old architecture.
Clever.
Then they introduced the fluorescent tag, so it would only attach to brand new proteins being built. They could watch the sparks of creation in real time.
And no-kutazole, the destabilizer, sent the glowing new proteins straight to the ends, the intercalated disc, right?
Yeah. Yep. And Taxol, the stabilizer, trapped them right near the nucleus at the sidewalls.
Visual proof. Unbelievable.
But if we connect this to the bigger picture, the clinical cases are the ultimate vindication.
Yes. Let's talk about the human tissue. They didn't just stay in rat models.
No, they looked at actual preserved human left ventricular tissue, non-failing hearts as the baseline.
And what did they see in the hypertrophic cardiomyopathy patients?
The ones with the thick walls. Just like the lab predicted, their cells were wider and their end cadherin was highly compacted at the intercalated discs.
Welded shut.
Welded shut. And the dilated cardiomyopathy patients with the failing stretched hearts. Their cells were longer and their intercalated discs were highly fractured and decompacted.
Wow. It's just amazing to me how this microscopic dance of microtubules exactly predicts the macro-level fate of a human heart beating in a chest.
It really is.
Okay, final recap for you. Bullet 1. Fluorescent tagging proved new proteins go exactly where the microtubule toggle dictates. Bullet 2. Human patient samples perfectly mirror the lab data. So think about your own body's adaptations. What other cellular highways might be secretly dictating the macrostructure of your organs?
This raises an important question, though, because if microtubule stability is the true upstream master switch, we aren't just managing heart disease symptoms anymore.
Right, no more just lowering blood pressure or using diuretic.
Exactly. Could the future of cardiology simply be a highly targeted tubulin tuning therapy, literally telling the heart to rebuild its own scaffolding?
It's incredible. We've gone from a single rad cell on a nanopatterned plate to the complex geometry of a human heart. It is genuinely amazing that the difference between a healthy heart and a failing one comes down to the rigidity of a tiny highway and a locked door at the end of a cell. So as you go about your day, think back to that self-remodeling house. Only now you know it's your own heart continuously tearing down and rebuilding at microscopic highways with every single beat. Thank you for joining us on this deep dive.